Namaskar. I am Ayesa
and I am Mehedi.
We are from Margram High School,
which is situated in Rampurhat 2 block,
District Birbhum and State West Bengal. In this project, our main objectives are we have tried to find out a local natural agent that can potentially eradicate any type of sewage harmful larvae.
Finding out of the larvicidal properties of calotropis lattice against our sewage-borne harmful larvae of Colognia sp, Chironomus sp and Culex sp.
We have tried to find out exact LD50 doses of latex.
Following proper reference, we have identified the test organism and Calotropis procera. We have collected the fresh latex of Calotropis procera and have done primary chemical and physical property tests.
Fourth stage instar of all three diphtheran larvae were collected in enough quantity in successive interval for qualitative and quantitative experiment at different times.
For qualitative test of each kind of species, fourth stage instars in a group of 10 were separated into two beakers. One beaker was kept as a control group with only 250 ml of water with no added latex. The other beaker contains 250 ml water and 5 drops of latex. Larval mortality were recorded at different time intervals.
5 experimental setups were taken along with a control for quantitative experiment which were done by only Culex species, in which containers of volume 250 ml only 100 ml pond water was added and 10 fourth stage fresh live Culex larvae were also added in each plastic container. Then serially 0.5 ml to 3.0 ml of fresh Calotropis latex were added in 5 containers except for the control. Larva mortality were recorded in specific time intervals. We have repeated the quantitative experiment for 3 times.
In the same way, Annables sp was tested as non-target organism in two 10-liter containers and the results were observed.
In all types of experiment, gradually change in skin color and softening larval body tissues have observed in increasing dose and increasing time of exports.
According to our quantitative analysis, we can say that fresh latex at relative concentrations of 0.50%, 1%, 1.5% and 2% against Culex sp have achieved 50% larval mortality at respective 120, 96, 72 and 48 hours.
But any dose less than 0.5 has failed or killed any larvae, and all of them has achieved maturity. All 5 appeal concentration of Calotropis fresh latex were useful against fourth instar of Culex species.
As almost all the Calotropis treated samples have shown the corresponding 50% and 90% mortality, we can able to calculate the LC50 and LC90 values for Calotropis fresh latex. For example, it is 2.75 and 2.375% of latex concentration respectively for 24 and 72 hours.
Calotropis latex appeared as a good potent larvicide. Experiment on non-target organism indicates that its future use must be restricted as a pond treatment agent because all 5 fishes in our treatment were died within 48 hours of latex exports.
We have worked together in this whole project from the beginning.
We are especially thankful to our guide teachers, mentors and our school for the all of our guidance and supports.